তাহলে আমরা শুরু করছি। Om Namah Bhagavate Ramakrishnaaya. Good evening, everyone. So, welcome to today's induction program for the newly admitted students of undergraduate microbiology in the Ramakrishna Mission Vivekananda Centenary College of the current academic year, organized by Department of Microbiology, Ramakrishna Mission Vivekananda Centenary College, Lahora. And we are very fortunate to have Dr. Shanta Dutta, Director and Scientist GA, National Institute of Cholera and Enteric Disease, NICET, Kolkata, for today's induction program. We are very grateful to you, madam, for giving us time from your busiest schedule for sharing your valuable insight views with our students and to motivate our microbiology students. So before going to Madam's talk, I would like to request Dr. Shekhar Pal, Assistant Professor, Department of Microbiology, Ramakrishna Mission, Vivekananda Centenary College, Lahora, to please introduce our speaker with audience and with us. Please, sir.
Thank you so much, Maharaj. Good evening to all. So I express my immense pleasure to have the opportunity to introduce our resource person today, Dr. Shanta Dutta, Director, National Institute of Cholera and Enteric Diseases, that is NICET, Kolkata, to our audience. Dr. Shanta Dutta is a clinical microbiologist and a medical research scientist. After graduated in medicine from Calcutta National Medical College, she completed post-graduation in medical microbiology. She joined NICET in 1994 and presently holding the post of Scientist G in the Division of Bacteriology and also the director of this institute. Dr. Dutta did her PhD in medical science from Kyushu University, Japan, and has more than 26 years of research experience in the field of enteric and diarrheal diseases. She is actively involved in field-based epidemiological studies as well as laboratory-based molecular studies with bacterial isolates, both clinical and environmental origin, with respect to their antimicrobial resistance marker, plasmid profiles, virulence markers, and sequence-based molecular subtyping, etc. She has acted as reviewer of a number of international peer-reviewed journals like PLOS ONE, BMC, Infectious Diseases, a Journal of Medical Microbiology, Antimicrobial Chemotherapy, etc. And also as a project reviewer of national and international funding agencies. She has published more than 250 articles in high-impact journals and various book chapters in the area of clinical microbiology, molecular epidemiology, and pathogenesis of enteric pathogens, water bacteriology, etc. She is the member of technical working group of the Global Task Force of Cholera Control Committee and participated in WHO meetings addressing the cholera control at the national level. She has worked as a principal investigator in a number of intramural and extramural funded projects and received various prestigious awards and fellowships in her career, like Lifetime Achievement Awards in 2017 of Indian Association of Applied Microbiologists, Vikram Sarabhai Memorial Award, Y.S. Narayana Rao Oration Award of ICMR, to name a few. Now it is over to you, ma'am, for much awaited deliberation at the induction program of our student.
Thank you very much for your very, very kind introduction. And I am so very much happy to get this invitation so that I can interact with the budding microbiologists. And for that, my immense gratefulness to Maharaj, who had contacted me, I think day before yesterday, and I readily agreed to his invitation. So this topic, it will be a kind of, I would be most happy if this would be a uh, like conversation kind of approach. But I don't think whether you all have the facilities of conversation. So I will give you in nutshell what is the. It is actually microbiology is a very extensive subject and it is very exciting subject as well. So I'll give you in nutshell those who would be pursuing your career with microbiology. What are the opportunities? What are the uh, I don't think there is any challenge, but you have to grab the opportunity and then you have to see any for any subject to grow in the subject, it depends on the individual. If you have the interest and if you want to know the detail of the subject, then I think you can, no one can stop you, and you can during in due course of time you can become expert in that subject. So with these few words, I start. I have a very, like I have made a presentation. So this is just for you, those the beginners who will be starting your journey with microbiology. Few points to note that there are several opportunities in this subject, and if you can delve into depth of this subject, then you can understand how exciting it is and how vast it is. uh, there are many areas, gray areas, where you can do research as well. So let me start with my presentation. So I think my slide is visible to you.
Yes, madam. Yes.
Yeah. So this is the subject as given by your professor. So this is the microbiology in higher education. uh, or if you like people like students like you who will be taking this subject as a professional career. So as I was telling you that microbiology, it is a very vast subject, and it is exciting, as well as it has many things, many research work or study you can do with this subject. It is ever growing subject. So this is the subject where we study microorganisms. Microorganisms, it may be bacteria, virus, fungi, protozoa. So these all come under together, we call it microorganisms. Those, it may be the microorganisms, it may be pathogenic, or it may be beneficial. Or beneficial microorganisms are those without without them we cannot survive. So those are the beneficial microorganisms. And there are some pathogens, and we are more concerned about the pathogens because they cause the disease. And some of the pathogens, they might have some potential to cause outbreak, maybe pandemic as well as pandemic, because we have all seen that COVID-19 pandemic, which started in 2020 March, and it has many lives have for this. It was a pandemic announced by WHO because it affected many countries, almost 130, 140 countries together, and many lives have been lost due to this infection. So this is one example how a virus can be so dangerous that it can have it. Virus, it is a very small object. It is less than zero point 0.4 micron, but it has the ability to spread across the globe and to take the lives of millions of lives. So you can consider this virus, how powerful it is. So I will not go into details of this, because if I start discussing microbiology, then there will not be any end to it. So uh, I will focus the opportunities what you can get if you study microbiology, either as your graduation course. You can, some who are interested to have higher studies, they can do post graduation as well as PhD for doctoral degree. This microbiology is a very, very interesting and big subject, where you can do lot of research to find out many more areas, many more findings which are yet to be revealed, or is not yet to be observed, yet to be revealed, and yet to be known to the world. So there are many opportunities. Just now I said that if you academic pathways, like, as you can go until doctorate, and after that also postdoctorate degree, you can be in academia, be a teacher, and you can teach the students and motivate them again. So this is one, of course, one opportunity which you can pursue. There are microbiology, it is many areas, like there are many disciplines in microbiology. Like, to name a few, this is one, medical microbiology, which deals with humans. Then environmental microbiology, which deals with the microbes of environment. Industrial microbiology, it actually, we study the, in industry, some microbes are very much important. In some are beneficial, and some of course can cause disease. Then food microbiology, agricultural microbiology, and there are many more areas where the microbiology has a special role to play, either as a beneficial role or as a pathogenic role, causing the diseases.
Madam, sorry to interrupt. Are you changing the slides? Because we are seeing only the first slide.
Yeah, I have changed the slide.
So that is not visible actually. We can view only the first slide.
Okay, okay. So I just, I'll please share. Hmm? I will. I'm just. I stopped sharing. Again, I'm sharing. You please tell me whether you can see, as I move, whether you can see the next slide. So this is the second slide.
Yes, ma'am. Now it is moving.
Okay. No. Now I come to third slide.
Yes, ma'am.
Is it moving now?
Yes, yes, ma'am. It is
Yeah.
moving now.
So research opportunities, I have already said, and teaching and academia, also industry roles. Microbiologists are essentially required for any industry where the microbes play a crucial role for the industry, like pharmaceutical industry, then biotechnology, then vaccine industry, healthcare. It is already I have spoken. Agriculture, food industry, then environment. There also a microbiologist can be consultant. They can play a bigger role for the entire public or the entire community, so that how to mitigate any disease which can occur by the microorganisms. From either it can come from animal sector, also very important. It can come from the animal, or it can come from the environment. So there is a big role to play for microbiologists. Then public health microbiologists, they can play a vital role in public health. Like uh, for all the diseases, infectious diseases, it can be, first thing you have to know what is the causative agent of that infectious disease. So for knowing that, you should have one accurate diagnostics in your hand. So diagnosis is the basis of medical microbiologists, and for that we should have one tool, diagnostic assay or diagnostic kit. So development of diagnostic kit is also one of the responsibility of the microbiologists, specifically those who are involved or associated with the research work. Then epidemiology and disease surveillance, infectious disease especially, is actually confirmed by a microbiologist by isolation and identification of the pathogen causing the disease. So to understand the disease dynamics as well as the treatment and management, the etiological, knowing the etiological agent is very important. Like I can give you one example. If the disease is caused by bacteria, then the patient needs some antibiotic for the treatment. But if it is caused by virus, like the disease is of viral origin, then there is no role of antibiotic. So if antibiotic is given, that is misuse of antibiotic, which can give rise to antibiotic resistance. So you can understand how important it is to know the etiology of any disease, infectious disease. Again, if the disease is caused by parasites, there is no role for antibiotics, or so antiparasitic agents need to be given for the treatment. And and after, with this treatment and management, appropriate treatment and management, only any infectious disease can be controlled, and the containment of the transmission of the microorganism is possible. So as I will give you some examples so that it will be easier. You don't know the subject yet, so if I give you some example, you can understand it better. Like when COVID-19, and I'll give the COVID-19 example because we are all witness of this vast pandemic, and we know how dangerous it could be. And when first COVID-19 pandemic appeared, then like the whole world uh, was in a condition that we don't know about the organism, the virus, how it is, how it behaves, how it can cause disease, what is the pathogenicity of this organism, then what is the treatment modalities, and how any patient is managed appropriately, what are the antivirals effective against this virus. Again, why the virus can cause two different disease conditions, like manifestation of the disease different in two different individuals, even if it is caused by the same virus. So these are all unknown to us. Here, how how much severe one organism can be in a host, sometimes it depends on the host factor. So in COVID, host played a very big role for the organism or for the disease to become severe, and even some of the patients, the final outcome was death. So that was also dependent on the host factors, host immunological responses to the pathogenic uh, organism. Then these are all the opportunities. So you can get some role. Hello? Yes? You can you can get some role to play as a microbiologist in some government as well as in some organization. Now, for even another big role to play for microbiologist is you have to become updated always with the latest knowledge or advancement in the field of microbiology, so that it is a continuous medical education. Like if you don't know the advancement, you will be losing the track, and you cannot be of use when the disease will cause any outbreak or any epidemic or pandemic kind. So, and for
Yes.
microbiology, one more very important area is field is networking or collaboration. So it is, any research is a teamwork. So a single person cannot do any research. So either national collaboration or international collaboration is very much the and only way to become successful in any research or gray area, to come to a logical conclusion or successful outcome. So this collaboration, it may be uh, with multiple stakeholders. Like it may not be so that one medical microbiologist will collaborate only medical field, environment microbiologist will collaborate only with the environmental experts, because any disease, there are three in a public health. Any disease, infectious disease, if it is caused by any agent, then three things are important. So it is known as triad of any disease. Like one is agent, another is host, and another is environment. So these three components are very important to control any disease. So if they, and now I think some of you might have heard one health approach. One health approach, it means you have to attack any disease from all aspect, like considering the multi stakeholders or the multi uh, disciplines, whatever like those disciplines, whatever involved in causing the disease. So like host, agent, and environment, uh, it may be dealt by different group of or disciplines of microbiologists. Then those microbiologists have to collaborate to target any disease for its control. And so I must say, with my experience in my professional career, I can say it is, microbiologist is a very rewarding and stimulating subject with several opportunities. And more so, it is the research or the scientific, those who have aptitude for research work or to know more details about the science, they will be benefited if they pursue this subject in their career. This already I have touched upon during my earlier that earlier speech. Like there are several disciplines of microbiology, which all of these are important for causing disease. And if one has to control the disease, all of these needs to be addressed. So this medical microbiology, then environmental microbiology, the name only will suggest like which area it uh, addresses. The microbiology, industrial microbiology, food microbiology, this is again one very important area. Like food, usually we get any organisms either by through our nasal route or oral route. So those which we get through oral route, usually those are the microbes which are present in food, water. Like we take those for our health. So food, if it is contaminated by microbes, then we can have many kinds of diseases. It may be, I have already said, bacterial, like typhoid, cholera, then shigellosis, like, or bacillary dysentery, and or if it is by these parasites, then uh, we can have amoebiasis, then several other protozoal parasitic infection, and also some tapeworm. Tapeworm, like it is it is usually people gets infected by fecal-oral route. So this is very important route for getting the infection inside the system of any individual. Then agricultural microbiology, which is also very, very important. Like human being, there are some microorganisms which can affect plants. So organisms are present in the soil, and also animals. Animals are also considered under this agricultural, like there are these are the areas which are dealt under agricultural council, like we have Indian Council of Medical Research. There is again Indian Council of Agricultural Research, and under this agricultural research all these are considered or studied. So plants also, some organisms which can affect plants, though those can cause damage to the plants. Again, I think all of you have already you know that some organisms which are very beneficial for the plants, and without those organisms the plants, I can't recollect exactly now what is that plant beneficial organism for plants, but those are very essential for their growth. And if the plants are devoid of those organisms, then usually there will be less growth. And then similarly in human being also, there are some beneficial microbes which are present in our intestine as well, usually on the mucous membrane, like intestine, then our respiratory tract, on the skin. And these are known as commensal organisms, which do not cause any disease to the human being, but they prevent the invasion by any pathogenic organism. So you can understand, they can fight with the pathogenic organism, and if uh, they won the fight, then the pathogenic organism cannot invade the human being. But where the pathogenic organism is more is stronger than the commensal, then only the human gets infection after by invasion of the pathogenic organism. Then immunology is another area, very, very interesting and intriguing area, a subject of microbiology. Like microbiology, microbes, when it is a foreign body, when it enters into a system, it gives rise to immune response. And this immune response is responsible for protecting the human being against further invasion or further attack of the same organism. So this is the basis of vaccination. Like when usually the vaccines are given for preventing any disease. Again, I'll give the example of COVID, like Covaxin or Covishield. All of us already we have taken the vaccine under the aegis of India government, and we have shown credentials in this area. Like this is the Covaxin is the India made vaccine, and when it is given, then it was observed that the case, it gradually the number of the cases it reduced, and now WHO has already declared that COVID is not a now it is not a public health, it is a disease. We still should be concerned, and we still should take appropriate safety measure, but WHO has declared it is not of public health pandemic. So it is a common disease now. Like it is not it has come down from its pandemic, that potential, to a very common disease of human being. So immunology, actually this is also very, very important and interesting area. One can always pursue. There are many things or many research work can be done in this immunology. It is not only the infectious disease, it is on the immunology is possible for virus, bacteria, any pathogenic organisms, or even the microbe what we have, the commensals. They are also responsible for uh, to some extent immune protection for the individual, like because we have those commensal organism, we have some innate immunity by which we can prevent the attack of some of the organisms. Then virology is another area. This can be, it is a part of bacteriology, virology, parasitology, fungus. So this is also, and virus is given much importance because most of the pandemics so far, what history has witnessed, it is usually caused by virus. So virus is given more importance because although it is a very minute organism, but it has the potential of causing pandemics. Then microbial genetics and ecology. Any microbes, when it is present either inside our body or in the environment, the milieu they have, those microorganisms, they interact with the environment. And if they get favorable milieu, then only they can multiply, which is true for environment as well as for the human body. And if it multiplies, if the microorganism gets adapted, the pathogen gets adapted to the system and it start multiplication, then it can cause disease. But if before multiplication, if it is attacked by our immune system and if the organism is taken away, then the person will not suffer from the disease. So this is very important for microbial ecology, how a microbe interact with the environment where it is present. So then microbial genetics also very important. Microbial genetics nowadays, whole genome sequencing facilities are available across many institutes of our country. And initially, the whole genome sequencing was expensive, but now it is not so expensive. And by this is a very important research, or I can say innovation, by which we can study the any microorganism in a deeper, in depth study can be done. So these genetic microbial genes, they are responsible for giving the foreign. Actually, you will come to know that genetic, by through genetic encoding, the microbes, they make their protein, which actually causes the harm to the uh, person in whom the organism has invaded. And these, with these genetics, we can understand the pathogenesis of the virus or the microorganism, and how the organism invasion can be controlled, or how the vaccine can be made from their gene, of target gene, or diagnostics. For making any diagnostic assay also, the target gene is important, to by which the organism can be identified in the system. In molecular microbiology, we all know the PCR, and there are PCR, then qPCR, then there are hybridization. So these are all advanced molecular techniques, and which are being used nowadays. In addition to conventional microbiology, this molecular microbiology is now popular to diagnose any disease or to detect the organism causing the disease. Public health microbiology, those microbiologists who worked in association with the public health, as I have spoken, that for surveillance it is important, and also vaccine trials or any other drug trial for infectious diseases. Microbiology is based on the report of the microbiologist only. The case, any disease or the case can be identified. And for a vaccine trial, identification of a case is very important. It should be confirmed case, otherwise you will not get the result, like safety or efficacy of the vaccine cannot be measured if if the microbiological result is confirmed, and it is well it is what should I say, like it should be confirmed and authenticated so that the disease can be identified. Then pharmaceutical microbiology, this is also important. It is important for safety and sterility. All the pharmaceutical products should be sterile. And quality control and quality assurance, these are two very, very important terms in microbiology. Like anyone can do microbiology, but if the quality is not maintained, then there is no use of that kind of microbiology. I have already said microbiology, work of a microbiology should be quality checked. It should be confirmed by any of the quality assurance or quality control activity. And if any lab is quality checked by quality control and quality assurance, then only I think many of you have heard accreditation of the lab. So that accreditation of the lab, it depends on how good the quality control of a lab is. So then you understand the importance of quality control. And this microbiology, these are all disciplines are interconnected, and the collaboration is very much required to get a good result. And there are several modern topics in microbiology, these are all interconnected again. So you will come across this terminology while studying microbiology. So like microbial genomics. So this is the whole genome sequence of in one microbe, so that you can understand what are the virulence genes present, what are the antimicrobial resistance gene present, and what are other genes. So all at a glance you can see if you analyze the whole genome sequencing of a microbe. Then metagenomics. Metagenomics, it involves the genetic material which you get of any microorganism which you get from environmental samples. And it does not need, like you need not go for conventional microbiology, but you just take the samples, like you can take stool samples or respiratory tract samples or environmental samples, water samples, and then just you extract the DNA and go for sequencing. So this will give, if through pathogen identification, if we go for sequencing, then we will get sequence of a pure organism, like only one pathogen or one bacteria or virus that can be sequenced. But when these metagenomics are carried out, then a whole lot of organisms, which are present in that environment, you will get a sequence of the entire gamut of organism. This is again important because some of the organisms are not culturable. All the organisms we cannot grow in the lab. There are some organisms which are fastidious, which do not grow in the lab condition. But if we go for metagenomics, we can, that means the gene sequencing of all the microbes, then you will come to know, then analyzing the genome sequence with the available data, you will come to know what are the microbes are present in the either clinical samples or environmental samples. This is important with respect to antimicrobial resistance, because antimicrobial resistance genes are not only present on the in the pathogen, like pathogenic organism. It is present in non-pathogenic organism, commensal organism, as well as in metagenomics, which we cannot grow. There also you will we have seen there also they have incorporated the antimicrobial resistance genes, and their close approximation with the pathogen, that gene can be transferred, the antimicrobial resistance gene can be transferred from microbe, like the commensal organism, to the pathogen. So here the source of that resistance gene is the commensal organism. So we must know what are the resistant genes present in the commensal organism or in the environment, and where the pathogenic organisms come in close with that organisms and get the genetic transfer of the resistance gene. Then system biology, this is also some computational or mathematical approach are linked to this system biology. Synthetic biology, so this engineering and biology together, you can design or construct some new biological components for the benefit of the mankind. But sometimes synthetic biology can cause biological warfare. I think some of you have heard, like sometimes there was one rumor that COVID-19, whether it is a synthetically produced, synthetically made, like scientists can make any organism synthetically engineered, so that either it can be useful to the mankind or it can be dreadful to the mankind. So this is important, you should have knowledge on this, and at least those who are researcher, they should do the research for the benefit of the human being. Then microbiome research, I have said that those microbiome are those organisms which are present without causing any harm to our system. So you can do some research on microbiome as well, because how you can have more benefit of those microbiomes, or how those microbiomes react with the immune status of any individual. Then CRISPR-Cas technology is another revolutionary gene editing tool by which you can modify the DNA sequence, and it has several applications in genetic engineering, antimicrobial therapy, so many other areas. Then microbial evolution and phylogenetics also very important. Like there are several microbial typing methods, molecular typing methods are available, by which you can understand whether these microbes, they belong to same types or they belong to various types. This is important to control the organism, because microbes of same type, they will can be controlled by single intervention or the procedure, but of different subtypes, they may not be controlled by single intervention. For those organisms of several same species but of several molecular subtypes, then you need to develop some kind of intervention. It is a multiple interventions may be required to control those organisms. Then microbial pathogenesis, we all know how one organism, when it enters into the system, how gradually it can cause the disease. This is very important with respect to developing new drugs, like the how you can target the organism with the drugs, so that the organism can be affected by the drug and cannot cause the disease. So this microbial pathogenesis is important in view in the wake of increased antimicrobial resistance. Antimicrobial resistance is the new silent pandemic, which is global pandemic, which is present mostly in the developing countries. And now we are in the post-antibiotic era. We do not have any antibiotic in our hand. If the bug, usually mostly in the ICCU or in ICU, if you get any organism of those patients, you will see multiple, they are resistant to multiple drugs, and there is not a single drug saved which can be used for treatment of those patients. So it is really, you have to think over it. And extreme environment also, this is also very important. Like some microbes, they grow in very hot, like even very cold environment or hot environment. So any organism which grow in the human being, they need an optimum temperature of 37, but or there are varieties of organisms who can be present in any environment. Microbial biotechnology is important for making vaccines. Like by bioengineering, only any vaccine can be made, and which is the final tool for getting rid of any infectious diseases or preventing any infectious diseases. and host microbes interaction also very important. Like I have already spoken little bit of it. So these advanced microbiology topics, you can understand how it represent the cutting edge research areas. So in all these, there are several scopes of research which contribute to our understanding of microorganisms and their diverse roles in various fields. So it can be of basic nature, then it is known as basic biology, or to applied nature like in biotechnology or in medical field like the clinics or in the epidemiology research. So as technology and scientific knowledge continue to advance, new exciting topics are constantly emerging within the field of microbiology, by which the microbiologist can have a in which microbiologist will have a bigger role to play to come up with few innovations which will be helpful to control the microbes in future. Thank you. So if you have any question, you can ask. I will try to respond.
Okay. Thank you very much, ma'am, your wonderful and encouraging speech. So now I request our students to please present your queries to ma'am.
Hmm? আবার কি হলো?
So I ask the students to present their query. Please unmute your microphone and ask the question.
Hello,
Ma'am,
Yes.
thank you for giving us a great insight into the different fields of microbiology. Ma'am, I want to know at what point of our career in microbiology we start to dive deep into the different branches of advanced microbiology, like you said about metagenomic study, microbiome research, and others.
Usually, first you have to go for the graduation and the post graduation. Like graduation and post graduation, you will come to know vast. It is, I said already, it's a vast area. So a glimpse of each of the areas, you will have some knowledge. But the real research work will start if you go for PhD, and there only you can choose your subject, which area you want to go in detail and which area you want to do research further. And there, I think there are options. So, and I always say to my PhD students that this PhD is the passport to the research world. Like in PhD, you will get to know how to do research, what are the like methodology, and what are the techniques, advanced techniques available, and exposure to high-end equipment. So after that, this 5 years, usually 5 to 6 years are required for getting a PhD, and during this time you will come to know the interesting part of this subject. Okay, so
Thank you, ma'am. Ma'am, I have another one query. Ma'am, what is the difference between MSc Biotechnology and MSc Microbiology in aspects of future research endeavors and opportunities?
these are all interlinked subjects. Okay, so if you have MSc Biotechnology or MSc Microbiology, for PhD, I think you all know now that after MSc also one can go for some job. Like it is not necessarily required that you have to go for research. only those who have aptitude for doing research, who get some interest after the new innovations or new areas of science, they can go for research. And actually MSc microbiology and biotechnology, you can say these are twin subjects, so it is interrelated subjects. And if you go for MSc microbiology, then also in a PhD you will learn biotechnology, and MSc biotechnology also will learn microbiology. So in before PhD there is not much difference if you whether you are taking microbiology or biotechnology.
Thank you, ma'am.
Ma'am,
Yes.
ma'am, so in PhD, what is the difference between biotechnology or microbiology?
Okay, so for microbiology, because I said these are very interconnected subjects, for biotechnology, microbes are required. Okay, so biotechnology means microbiology, it deals with the microbes. And when you are engineering, you are doing some kind of uh, experimentation. Like I can tell you, biotechnology, bioengineering or biotechnology, where the organism is considered as a base or as a vector to do some kind of genetic manipulation or modification, that comes under biotechnology. So it is not necessarily a microbiology PhD will know only microbiology and no biotechnology, because doing research you have to do some kind of biotechnology. Only microbiology you cannot do research. Again, if you do not have entire concept of microbiology, you cannot do biotechnology. So don't consider these two subjects are very different. These are, one is complementary to another. And biotechnology is mostly, it is the, you can have um, MSc biotechnology as well as uh, some engineering, BTech biotechnology is also there. So it is more that manipulation of the genetic manipulation. It is you do it in biotechnology, but until and unless you have some knowledge on which you will do the manipulation, this microbiology world is a big world. You should have some knowledge on it. Otherwise, biotechnology you can do in any of these, either parasite or bacteria, virus, any of the organisms, biotechnology is possible.
Ma'am,
Ma'am,
thank you for your emerging speech, ma'am. If I want to do a job in after MSc, so what should I do then?
After MSc, you are in MSc, BSc microbiology, right? You are
Yes,
doing
ma'am.
BSc microbiology. So if you go for MSc microbiology, there will be certainly one chapter or one, you say some your subjects, biotechnology. Certainly there will be one. So you can take your specialization in biotechnology. But microbiology is a bigger subject, and a portion of it is biotechnology. Biotechnology is rather, I can say it is a subject, but it is a kind of technology. So whether you understand or not, it's a kind of technology. So technology can be applied to many things, like many, I said that microbiology entire area you can apply biotechnology. So if I prefer, you should go for, then your, if you want to get a job, then this MSc microbiology have a bigger area, like in food, environment, everywhere my microbiology is required. And if you take biotechnology as your special area, you will get to know in a better, in a more um, like not in a research mode, but at least in an expanded mode, you will get to know the biotechnology. So that will fulfill the biotechnology area and the microbiology, because you have studied microbiology, so you will know the microbiology also.
Thank you, ma'am.
Ma'am, I have another question that what is the scope behind biomedical sciences
What is
in biomedical sciences? And what is the scope about this?
biomedical science? What we are doing, we are all doing biomedical science. All research related to medicine, it comes under biomedical science. So whether so some, like, if you, even if you go for some, maybe some veterinary research, but you are doing with the organism which can cause disease to the human being, then it comes under biomedical science. Like any science related to medicine, like human being, and on other aspect it is related to biology. So those together is known as biomedical science.
Okay, ma'am, thank you.
Good evening, ma'am. I had a question.
Yes, please.
Ma'am, you were elaborating on the topic of antibiotic resistance. Ma'am, actually, I had read a paper about the survival of Mycobacterium tuberculosis inside stem cells. That can be a cause of antibiotic resistance also. So, ma'am, I want to know that is only consuming a lot of antibiotics the main cause of antibiotic resistance, or can the survival in stem cells also be a potent cause for this thing, because the pathogen can then hide from the host immune response?
Of course, that having indiscriminate intake of antibiotic, it cause antibiotic resistance. And but if any what you are telling, that survive that Mycobacterium tuberculosis, presence of Mycobacterium tuberculosis in stem cell, but I would rather say that presence of typhoid bacilli, it's much more simple organism in gallbladder, which can cause chronic cholecystitis. So this, if it is present, usually it is present if the uh, person does not take the full course of the medicine of and half the way, or half course of the medicine, or the dose also, it is not the full dose of the medicine, then what happens, the organism, it it becomes exposed to the sublower concentration of the antibiotic. That concentration, it is not bactericidal, neither inhibitory. So there it sustains, the organism sustains, and gradually it develops resistance, because we all know that survival of the fittest. So in that case, organism also start changing its machinery so that it can stay in that environment, it can live in that environment, and then it has to become resistant in that environment of antibiotic, low dose of antibiotic, and that can cause resistance. So what you are we have mentioned, both the factors are important for causing antimicrobial resistance. Like indiscriminate use of antibiotic, when antibiotic is not required, I am taking antibiotic. Again, when antibiotic is required, I am not completing the neither the dose nor the duration. So both these factors are important for development of antimicrobial resistance against the organism for which you are taking the antibiotic.
Thank you, ma'am.
Good evening, ma'am.
Yes.
Ma'am, recently some studies say that bacteriophage is an effective for certain antibiotic work. It works like that. So, ma'am, how is like the future prospectus of its research?
Yes, you are right. Like, I'm very happy that you are very, all of you have that interest and you have studied also. So this phage is another, actually this, you will come to know this bacteriophage is a virus kind, it is a virus actually, the phage. If you structure, if you study, it resembles virus, but it needs some host. Virus cannot survive by itself. It needs a cell for its replication. So similarly, the bacteriophage, it needs the cell specific to that bacteria. Like suppose bacteriophage, vibriophage. So it needs vibrio cholerae cell for its multiplication as well as survival of the phage. So there are two types of phage. One is lytic and another is lysogenic. So lytic phage, what it causes, like it when it enters into the cell, it multiplies, and then after repeated multiplication, when the number of the phages become heavy, then it causes lysis of the cell, because it replicates using the host machinery. And so when the replication, it reaches to the highest point, then it cause bursting of the bacteria. So this is the principle which is used for treatment of any bacteria, any bacterial infection with the respective phage. So, but this is a very um, promising area, I must say, because nowadays most of the antibiotics, whatever antibiotics we have in our hand, almost all the bugs, they are showing resistant to that antibiotic. And it is very much unfortunate that after the invention of 1940 penicillin by Alexander Fleming, there are certain derivatives, some derivatives have been invented out of the penicillin, but no new antibiotic after that has been invented. So in view of that bleak area of invention or innovation, if this bacteriophage can be used for the treatment, no doubt, because it is another biological material we are using for treating the infection. But we have to several steps we need to go, because this is a virus, as I have said. So it has to be, and we are, if it can be used for treatment, then we are introducing that phage, the biological material, into the system. So you can understand that it has to be purified. It should be very specific to the pathogen, like it should not cause lysis of any other organism. So there are a lot more work we have to do. Some international, not like some international organizations who have already started this phage isolation, characterization, and uh, the treating some of the infections with this phage therapy. But one is in Russia and another, I think, is in US. But until now, this is very expensive, and it may not be so convenient for the developing country where the infections are much more common to follow the phage therapy. So we have to yet to do many more steps to get to that point where, of course, it can be used, and where the phages can be used for the infection caused by the same bacteria.
Okay, ma'am. Understood. Thank you, ma'am.
So, okay, thank you, madam. So, with this, we come to the concluding moment for today's induction program. So, on behalf of our college, on behalf of our department, and also on behalf of our all faculty members, I extend a really heartfelt gratitude to you, madam, for spreading so much time for your busiest schedule to grace the occasion and to enrich us. So I also thankful to you, madam, for your wonderful talk and interactions with our students. And today we had an opportunity to hear your thoughts, your inside views, and we will be surely it is going to be encourages for us, for our students, for their various future progress. And we will hope that they will definitely enlighten us again. And we invite you, madam, to please come and visit our college, our department, and as well as to interact our students to motivate them in future. Thank you, madam.
Thank you.
Thank you
Thank
very
you.
much.
Thank you very much.